muted during the session for the seamless learning experience on Zoom platform. Participants are requested to keep their videos off on Zoom platform. Please do not raise your hands. All are requested to post their questions in the chat box on Zoom or YouTube. The questions will be taken up after the session during question and answers round. Instruction to participants. The participants will be muted during this session for seamless learning experience on Zoom platform. The participants are requested to keep their videos off on Zoom platform. Please do not raise your hands. All are requested to post their questions in chat box on Zoom or YouTube. The questions will be taken up after this session during question and answers round.
YouTube live also there, no, sir?
Yes.
YouTube live.
Yes.
The same link, na? Camera in that camera, right? You party. Madam.
Dear participants, please listen to the instructions. The participants will be muted during this session for seamless learning experience on Zoom platform. Participants are also requested to keep their videos off on Zoom platform. Please do not raise your hands. All are requested to post their questions in chat box on Zoom or YouTube. The questions will be taken up after the session during the question and answers round.
Sir, Zoom on the paid version, sir. Sir?
Paid version అండి.
Okay, sir. Trial version అందరి క్లారిటీ రావట్లేదు సార్.
No, paid version.
ఇది paid version సార్.
Okay, sir. ఏ చెప్పండి. How many members are joined, sir, so far?
So far 65 sir.
65.
Okay.
65, I think up to 100 we are having chance, and then the remaining people they will go to YouTube sir.
And share screen கொஞ்சம் enable பண்ணுங்க சார். நான் just PowerPoint. Share screen எல்லாருக்கும் மாஸ்க் கம்பல்சரியா இருக்கு. Tuesday also session is there, sir.
Which kind is Shalom sir?
Tuesday actually we are organizing from society, sir, computer-aided drug design.
Okay, from your society you are organizing.
Yeah, society organizing.
Okay.
And Dr. Ashok Sharon from Birla Institute of Technology, Ranchi.
No, if you send the link to me, limited members from our college will join, sir.
At least chemistry, but because he is from USA, sir, studied from USA.
No. I will ask my chemistry colleagues to join, sir, at least.
Okay.
It's better, sir, because they will have some exposure, sir.
Sharing disabled. Sharing disabled panna, just I will check. So today I am going to discuss only on HIV, sir. HIV drug discovery development.
No issue, Selvam. Just
Yes,
it is
we
also
have
antiviral category only, na?
Yeah. We have vast experience with HIV, sir. The new we are working on molecule for HIV as well as new mechanism also.
But now my sincere appeal is now this pandemic is there, na? That is also viral pandemic only.
Already I given 5-6 talk on COVID drug discovery. So I sent the PowerPoint. Okay.
But my sincere appeal from all of our Andhra people or other people also, please start work on that also, sir. Actually, that is better.
Yeah. Actually, this is a common methodology, sir. How to understand the life cycle, what is the molecular targets, how we can arrest, how we can apply the technologies, screening techniques, molecular biology, bioinformatics, chemistry.
Okay. Okay, Selvam.
Yes, sir.
Everything will be recorded in YouTube. We will talk personally, no problem.
Okay. Actually, I put one title given as a antiviral drug design discovery and development. Among that, we are going to discuss very important aspects, sir.
Indra. Okay, right. అప్పుడు పెట్టమన్నారు presentation. Okay, right.
Hello.
Selvam.
Sir.
Just your presentation, just for a moment, please stop. First, my colleague
Okay.
brief introduction about seminar, and then I will introduce you. Then you can start. Okay.
Okay.
Thank you.
Hello, sir. Please come out of your screen.
Selva,
Yes, sir.
that screen, your presentation.
Can I bring me, sir? Okay.
Just few, no. Few minutes if you stop now, then right. నేను 2 minutes మాట్లాడతా అండ్ ఐ విల్ ఆస్క్ సెల్వన్ టు స్టార్ట్ ద ప్రోగ్రామ్. అంటే ఓకే చెప్పారు మాస్టర్ చెప్పారు, విల్ ఫాలో దట్. ఓకే.
Uh, 10 o'clock అయింది సార్, స్టార్ట్ చేయమా?
స్టార్ట్ చేస్తున్నా సార్.
Good morning, one and all. Myself, Dr. G. Vijay Kumar, Professor of KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada. Welcome you all for today webinar on recent advances on antiviral drug discovery and development, which is organized by IQAC Cell, KVSR Siddhartha College of Pharmaceutical Sciences in association with Antiviral Research Society. It's my privilege to speak a few words about our prestigious institution. Kommareddy Venkat Sadasivarao College of Pharmaceutical Sciences, KVSR SCOPS, was incepted during the academic year 1993-94, and it is the first private pharmacy college started in the combined state of Andhra Pradesh. The college is sponsored by Siddhartha Academy of General and Technical Education, SAGTE Vijayawada, which is a visionary society running several institutions. The wide range of institutions run by our SAGTE includes medical college, dental, pharmacy, engineering, and education, management, law, and arts. The college is affiliated to Krishna University, Machilipatnam, and is approved by AICTE PCI. The college offers B Pharmacy, M Pharmacy, Pharma D, and Pharma D postbaccalaureate programs. Our college is a approved research center for pursuing PhD under Krishna University. For the first time in Andhra Pradesh, Pharma Innovation and Incubation Center SPIC was established under management of the Academy of General and Technical Education. At KVSR Scopes, several DST and other sponsored collaborative research projects are under process. The college campus is located in the middle of Vijayawada city with all advanced amenities. KVSR Siddhartha College of Pharmaceutical Sciences is committed to the education of pharmacists with higher ethical and professional standards. The college has excellent academic atmosphere, culture, discipline with best infrastructural facilities such as building, laboratories, library facilities, student hostel and playground, etc. Now I request our beloved principal sir, Dr. G. Devalra Garu, to give a brief note on today's webinar and also introduce today's guest speaker, Dr. Selvam. Before that, a small instruction to the participants. The participants will be muted during this session for the seamless learning experience on Zoom platform. Participants are also requested to keep their videos off on Zoom platform. Please do not raise your hands. All are requested to post their questions in the chat box on Zoom or YouTube platform. The questions will be taken up after the session during the question and answers round. Thank you. Sir, please, sir.
Thank you, Dr. G. Vijay Kumar. Good morning to all of you, and a very good morning to all the participants in the webinar. First of all, I want to congratulate our IQAC cell headed by Dr. Vijay Kumar for organizing this webinar on recent advances in antiviral drug discovery and development. So for that, we have selected a very right and apt person who is none other than Dr. P. Selvam. So Dr. P. Selvam, he has completed his B.Pharm, M.Pharm and PhD from a prestigious institute in Tamil Nadu, that is Madurai, Madurai Medical College affiliated to Dr. MGR Medical University, Chennai. Totally, he is having 17 years of teaching experience. In that 10 years, specifically, he is having research experience also. And based on his research work carried out, he has published 75 papers in national journals and 25 in international journals. And he has presented 50 papers in national conferences and 50 in international conferences also. After that, his field of specialization is mainly pharmaceutical chemistry. In that, he is mainly interested in antiviral research. For that, he has started his own society, that is Antiviral Research Society also. For that, we are very happy and we want to congratulate Dr. Selvam. After that, he has guided so far two PhD candidates for their PhD work and 25 candidates for their M.Pharm work. And he has published several books. He is having national and international patents also. And he is having professional membership in APDI, ISAR, ISNS, NMRS, NABS, IAMPV. After that, he is having very good consultancy activities with Noni Biotech Private Limited, Chennai, worth of 11 lakhs. And he got several awards also for his credit. The first and very important is DST Boycast Fellowship Award in 2007. And then Noni Outstanding Research Scientist Award in 2011. Fellow of National Academy of Biological Sciences in 2012. Next one is Noni Service Excellence Award 2013. Fellow of International Society for Noni Sciences in 2015. He has, to his credit, having a grant worth of 10 lakhs from Noni Biotech Private Limited, Chennai. And he is having interaction with the various professional institutions like National Institute of Immunology, New Delhi, IIT Madras, Delhi University, Dega Institute, Belgium, NCI, NIH from USA. With this brief introduction regarding the my good friend and eminent scientist, who is none other than Dr. P. Selvam, and today he is going to deliver a talk on recent advances in antiviral drug discovery and development, and I think he is going to concentrate on HIV drugs, I think. So thank you very much, Dr. Vijay Kumar. for your kind words. And now I request Dr. Selvam, the speaker, to start his presentation. Thank you, sir.
Thank you, sir. Switch off my presentation. Slides. So can we start, sir?
Yes, sir.
You can start, Selvam. Start, please start. Please start.
So show the slides, sir. So on behalf of Antiviral Research Society, I welcome you all. So I express my sincere thanks to our beloved principal sir and management for accepting the invitation. So it's currently right time to do the research on antivirals. Pharmacists, we are the pharmacists, we are the maker of the drugs, administrator of drugs and dispensing the drugs. So currently, like SARS coronavirus, number of many viral diseases appear, which is pathogenic against the human being, like swine flu. We have many chapters of experience with the swine flu, dengue. Slide. Dengue, chikungunya, like this, many viruses will appear. Most probably, most of the pathogenic viral disease which is caused by the RNA virus. Generally, the viral may be two categories, DNA virus and RNA virus. So as a pharmacist, currently we want to focus on the development of antiviral drugs, vaccines and prophylaxis and herbal-based medicine for the treatment of this coronavirus and other viral diseases. Today we are going to discuss about the recent advances in antiviral drug design, discovery and development. So we know that India is one of the leaders for the HIV/AIDS. India, our total population, 0.5% people are infected with the HIV virus, which causes AIDS. So HIV virus is an intracellular replicate parasite. It's a very typical virus. So we are going to.
Selvam, sir, I think presentation is not there, appearing.
Can you share your?
Okay, okay, sir.
Yes.
So,
No.
yeah, slide, sir, slide, second page. So today we are going to discuss about the antiviral drug design, discovery and development. So for the purpose, well-established HIV virus. We are working on the HIV virus past 20 years from 2002 to we are working on the HIV virus drug discovery and we are working on new developmental targets. So HIV virus is one of the major problem. It also belongs to the RNA virus. Already I stated that viral disease may be two categories, DNA viruses and RNA viruses. Currently, all human pathogenic disease which is caused by the most probably RNA virus like a swine flu, Nipah virus, dengue virus, chikungunya virus, and recently for SARS coronavirus also. We had the experience of SARS coronavirus in the 2003 and 2012 we have the MERS virus, Middle East respiratory. Now we have the SARS coronavirus 2 and the COVID virus. So in order to understand the antiviral drug discovery process, we want to understand the life cycle, or we want to understand genomics, what is the validated molecular target and screening techniques, and we have to apply the chemical biology as well as the new chemical entities and natural products to discover new molecule. Today we are going to discuss about how we have designed the drugs for the HIV virus, because HIV virus is one of the incurable disease. So we need more attention towards this virus. So they are recently the 3 persons which shared the Nobel Prize for physiology or medicine due to the discovery of HIV virus, human immunodeficiency virus, which causes AIDS. So in order to understand drug discovery, antiviral drug discovery process for any viruses, is a common approach is we want to understand what is a virus, what is its life cycle, what is its genomics, is a very, very important part of the pharmacist or pharmaceutical sciences. Already I told you, HIV virus is a RNA virus. Recently, COVID virus also RNA virus. This RNA virus is having this structure of virus. We want to understand the structure of virus, then only we have to know the what is the well-dated molecular therapeutic target. So, like other viruses, they have the spike protein, glycoprotein for 120 as well as glycoprotein 41. So this spike protein, which is mainly responsible for the antigen antibody reaction and vaccine development. Then inside they have the membrane, envelope protein, nucleoprotein, as well as the number of enzymes are there. So these are three-dimensional structure of HIV virus. So this is the genomics. As a pharmacist, it's called pharmacogenomics. So we want to understand the genomic aspect of the virus. The genomic aspect of the virus, then only we have to know what is the important enzyme which involved in the replication of the virus. So here we have to know that middle portion have a PR, RT and integrase. As a protease, reverse transcriptase and integrase. The protease called as PR, reverse transcriptase called as RT and IN called as integrase. So these three. Like our India Hindutva, like this Vishnu, Shiva, and Brahma. These three gods are very, very important. Similarly, this HIV virus development, multiplication, these three is very important. Integrase protease for protein synthesis, reverse transcriptase for conversion of RNA and DNA, and integrase for the integrase. So these three enzymes are the validated therapeutic target for the discovery of new molecules. Currently approved ART, antiretroviral therapy, and heart therapy, highly active antiretroviral therapy. So this based on the inhibition of these three viruses. Apart from that, they are having the GP 180, 120 and for GP 41, that is called membrane protein, which is responsible for the binding. So the new class of inhibitors called as entry inhibitor. The inhibitor which prevent the viral entry. These are which is attachment of GP 120 with the CDP4. And apart from that, the virus having number of cellular accessory protein, that is called as regulatory protein like CA, PA, NC like this, and VIP, virus infective factor. So in order to understand the virus as well as genomics is very, very important for the drug discovery process. So in order to understand the molecular therapeutic targets, we want to know the life cycle of the virus. We want to know the life cycle of the virus. Here, this is a single slide which explains the life cycle of the virus as well as molecular target for the HIV virus. So here, for the replication of the virus, first the HIV virus will attach to the HIV virus which contains the GP120. It's a glycoprotein which is attached to the CD4 receptor.
Selvam sir?
Yes, sir.
Please put that presentation in full screen.
Full screen, huh?
Full screen.
So actually, we converted in the PDF, sir. So that's why.
Oh, PDF. This is so.
Okay, that's why, sir.
PPT, is it PPT or PDF, sir?
It's a PDF, sir.
PDF scroll out.
Okay, sir. Can I continue?
You can continue.
So here, very, very important point. First, the virus which contains the glycoprotein 180, which is attached to the CD4 receptor. Once the virus is attached, then which is undergo the endocytosis, the outer membrane is fused with the cell membrane, then will go inside the lysosomes. The outer coating can be removed and viral RNA along with the enzymes, reverse transcriptase, integrase, and the protease can be released to the cytoplasm. Once the viral RNA is released, then viral RNA is converted to cDNA with the help of the enzyme reverse transcriptase. It is, I already told you, this HIV virus is a very peculiar virus. In other viruses, DNA converted into RNA is a transcription, but it's a reverse process. That's why it's very complicated for developing the vaccine as well as therapeutic, novel therapeutics. So here, HIV RNA is converted into cDNA, clone DNA, by the enzyme reverse transcriptase. So most of the drugs nowadays are targeted for inhibition of reverse transcriptase. Once the reverse cDNA form, that can be entered into our genomics, our nucleic acids. Once they enter into our nucleic acid, the integration is a process which is aided by the enzyme integrase. So currently, integrase is an attractive therapeutic target for discovery of new molecule. Once it is integrate the host genomes, they will convert the human DNA into the viral DNA. Once viral DNA is converted, automatically the viral DNA once again is translated to the RNA. They come to the ribosomes. Ribosome is a cellular machinery which produces the viral as well as human protein. That is called as a translation. Once viral RNA formed, translation formed, protein is the size, all the protein assembled together, they form the new virus and then released. Here, 5 steps are very, very important. Viral attachment, that is called as viral entry. Drugs which inhibit the viral entry, and then RT, reverse transcriptase inhibitor. You know the process of transcription. A DNA converted into RNA is called as a transcription. RNA converted into protein is called as a translation. This is a reverse transcription. Generally, DNA converted into RNA. Here in this case, RNA converted into DNA. So, with the help of the enzyme, reverse transcription. Reverse transcriptase is a protein which converts the viral RNA into cDNA. Then once the cDNA formed, the cDNA can inserted into the host genomes. Host genomes with the help of the enzyme integrase. Integrase is the enzyme which is present only in the virus. Once they integrate in the human genome, they will convert the human DNA into viral DNA. Once viral DNA formed, viral DNA converted into RNA. The translation, RNA will come out, will go into the ribosomes. They produce the proteins. Protein all together matured and then released. Then one virus can enter, number of virus can be released. That's why they will destroy all the host cells, CD cells, lymphocytes. Because virus already told you it's an intracellular obligate parasite. They need specialized atmosphere to grow. And also they used the number of human protein for their multiplication. So that's why virus called us our ancestors. Here from that life cycle, we have to understand attachment and the reverse transcription, integration, protein synthesis, maturation and release. So here the single slide will explain that the viral entry inhibitor, viral reverse transcriptase inhibitors, viral integrase inhibitor, virus protease inhibitor and release, the maturation or transactivation inhibitors. So this aspect we are going to focus mainly on the integrase because reverse transcriptase and protease also present in the human cells also is a commutative. So viral integrase, which is helped to integrate the DNA from the cytoplasm to nucleic acid, which is present only in the virus, not in the human. So and also it is a very unique to the virus, very attractive therapeutic targets. Based on the life cycle and validated therapeutic target, that is called druggable targets. If you simple example, if you want to capture one country, we have attacked by aerial way, by flight, by land side, by the seaside, by the naval. So a combined attack will capture the country easily. Similarly, not to remove the virus from or clear the virus from the human being, one will prevent the viral entry, that is called as entry inhibitor. One will prevent the fusion with the cell membrane, fusion inhibitors. One will inhibit the enzyme reverse transcriptase, RT inhibitor. One will inhibit the integrase. One will inhibit the protease, one transaction. Minimum among the 6 approved combinations, minimum 3 combinations is very, very important. Currently used ART, antiretroviral therapy, one which inhibits the RT, protease and integrase. So combination of these 3 drugs which clear the virus our human host system. So these are the validated attractive therapeutic targets. Number of drugs which is approved for treatment of HIV/AIDS by the FDA, Food and Drug Administration, USA and WHO, because of the viral fitness, site-specific mutagenesis, and viral drug resistance, all the drugs causes resistance to the virus. So, because as per the Darwin's theory, survival for fittest means the organism changes the amino acid to the active site that they make them inactive against the antiviral agents. So here, with the reference to the previous life cycle, I will list out some of the drugs which is approved by the FDA. Entry inhibitor, enfurides, marovirac, and visirolac, and nucleoside reverse transcriptase inhibitor like zidovudine, lamivudine, titanosin, nucleoside reverse transcriptase inhibitor like tenofovir, adifovir like this, and nucleoside reverse transcriptase inhibitor like a non-nucleoside small molecules like nevirapine, dilarvudine, efurins. So these are the small molecules which inhibit the reverse transcriptase. Then protease inhibitors. Protease, after the transcription, the RNA will reaches the ribosomes. The ribosome, inside the ribosomes, they will prepare the viral protein. Any RNA, either is a viral RNA or a protein, a human RNA which enters the ribosome. Ribosome is a cellular machinery in our system. they will produce respective proteins. That is called as a translation. So here, drug like tenofovir, sipronavir, ritonavir, intenavir, nalfenavir, tenofovir drugs. Recently, on happy news, HIV protease inhibitor, which also inhibit the SARS coronavirus, COVID 2019, 3CL protease, chemotrypsin like a protease. So, lopinavir and intenavir combination is recommended for treatment of SARS coronavirus. And last one is integrase inhibitor. Integrase is a very crucial enzyme for HIV replication. Today, our presentation, we are going to discuss about how to design the integrase inhibitor. So, these are the FDA approved drug for treatment of HIV. Then we have to understand the genomic aspects. Already I told you it's a protease, reverse transcriptase, integrase. We are going to focus mainly on the integrase inhibitor because integrase is the enzyme which is present only in the virus, not in the human. So there's more chances of getting more clearance of virus than the other things. So this is a structure from the cytoplasm that pre-integration complex, PIC. Pre-integration complex means viral HIV integrase and the HIV DNA, cDNA and the post protein. This is called as LEDCF, lens epithelium derived growth factor. It's a post protein P75 transcription. So these three, they will form the PIC, pre-integration complex. They will insert the cytoplasm to nucleic acid by pores. It's called pre-integration process. Once the DNA can enter, they will integrate with the human DNA. They will integrate with the human DNA. Once they integrate with the human DNA, the replication will be started. So 3' portion of viral DNA will bind to the 5' portion of the human DNA. So this is called as 3' processing. Similarly, 5' portion of the viral DNA will bind to the 3' portion of the human DNA. So this is called as strand transfer. These two, like a Kyer industry, the two threads are joined to form the Kyer like this. One end of the viral DNA, one end of the human DNA will die. So similarly, there is in the integration process, there is a 2 steps: 3' processing, strand transfer. So in the second picture also will explain that. So this is a sequence. Once they are cytoplasm, they are integrated. Then through nucleopore, it can be integrated. So the integration is an important process. If you arrest the integration process, we can effectively control the virus replications. And we already told that virus are our relations, our ancestors. So why we are telling like this? For the multiplication of virus in the host cells, they need number of human protein, host protein for their multiplication. So we selected few of the protein. Is a very, very important for the virus multiplication. Is called as a P75 transcriptional cofactor. That is called as LEDCF, lens epithelium derived growth factor. It's a post protein which is present in the, actually 2 proteins are there, P51, P75. P75 is a protein. So we studied that inhibition of this P75 in the HIV will have the no replication in the cells. So this is an electron microscopy structure. How the box inside the DNA is a viral DNA. You see how that viral DNA can be nicely integrated with the host DNA, host genomes. So this is 2 steps are there: 3' processing and strand transfer. I am explain this process. These 2 are very, very important for the drug discovery and development process. So this is a nice electronic picture how the viral DNA can be integrated with host DNA. So we want to understand the integration mechanism because we selected only the integrase for the drug discovery process because HIV integrase is a crucial enzyme for multiplication which is present only in the virus. So if you select the druggable therapeutic target or validated therapeutic target, so and also they have a less resistant, drug resistant, chance to drug resistant. So for that purpose we selected this 2. So here it should be very clear. Once the virus can attach to the cell surface, then reverse transcripted, then it can be integrated with the host genomics through the nucleopore. For nucleopore, they require number of human protein also. Here there is a 2 processes are very important: 3' processing and strand transfer. So this is a sequence of event how the PIC, pre-integration complex. The pre-integration complex consists of 3 components. Number 1, like it's a LEDZF. It's a host protein. It's a small protein present in the human, and it's a HIV DNA. It's a cDNA, cloned DNA. Plus, there are two ball-like structure is called as HIV integrase. So these three compounds they will form the PIC. It's a pre-integration complex. After a virus attached, their outer coating is removed. Their RNA is released. RNA converted to DNA. DNA are combined with the integrase and the LEDZF. They will transport from the cytoplasm to nucleic acid. One copy of DNA can transport from the cytoplasm to nucleic acid. Thousands of copies of viral DNA can be generated by the process of integration. So they will nicely go via the nucleopore. Main number of proteins, transport proteins are available to facilitate the transfer of these PIC from the cytoplasm to nucleic acid. So this is a process. Here we are mainly focusing on the LEDZF and interaction with the integrase. Already I told you the virus is an intracellular obligate parasites. They will replicate only specialized cells, not all the cells. For coronavirus means lung cells. Hepatitis virus means liver cells. HIV virus means CD4 cells or lymphocytes, because virus is a obligate intracellular parasite. They need specialized atmosphere for the multiplication. So for the process, and also a very important thing, the virus they need number of host protein, cellular protein for their multiplication. We have taken the T cells and add the HIV virus and for the incubate for 5 days for multiplication. This effectively multiplicate and it has a side of the effect. So we for the assessment of host protein is very, very important for the virus multiplication. We removed the LEDZF, lens epithelium derived growth factor, that gene from the host cells by the gene deletion, or we make them silencing or yeast hybridization methods. We removed the gene P75 genes and we add the HIV virus. We found that HIV virus cannot replicate. That is, they are not produce any cytopathic effect. From that evidence, we proved that P75 is a very, very important binding partner for HIV integrase. HIV integrase is a viral protein, will bind with the LEDZF, lens epithelium derived growth factor, host protein. They form the complex. Then only they will transfer the DNA from cytoplasm to nucleic acid. So we are analyzed the genomic analysis while I am doing the postdoctorate in Belgium. We are genomic analysis that the cells which are having two different type of P51, P75, P52 and P75. Both belongs to the lens epithelium derived growth factor. Is a host protein, human protein, which is present in the cells for their cell apoptosis and the stress condition. So we identified that P75 and P52 genomic analysis only the P75 have the IBD, that means integrase binding domain, whereas P52 does not have any IBD. This IBD is a site where integrase, HIV integrase is bind with this sites. So here with this genomic aspect of HIV integrase, the HIV integrase have N terminal and C terminal and the central core domain, which contain the TT2 aspartic acid and tocoglutamic acid, which is a catalytic triad. So this is central core domain will bind to the IBD. So we will perform the protein-protein interaction studies. We will confirm that among the two proteins, P52, P75, only P75 having the integrase binding domain. Next, we will go for discovery process. This is previously we discussed about the life cycle, genomics, therapeutic target. Now we will discuss about the anti-HIV drug discovery. So I already told you each virus belongs to the specific category of cells for the growth. For liver cells, HG2 cells for the jaundice, HPV, HCV. Similarly, viro cells for the SARS coronavirus. So cell which is specified for the growth concern. That means like a 5-star hotel accommodation, the cells they need intracellular obligate parasite. They need specialized condition for the multiplication. For commonly used cell culture for drug discovery process, it's a T cells, it's a lymphocytes. So here we have taken the T lymphocytes. We analyzed thousands of compounds in the cell line. Thousands of compounds in the cell lines. We have the T cells, MT4 cells. This is a C-type adult T cell leukemic cells which infected with the HIV1. It belongs to the HTLV3B. It's a human T lymphocyte leukemia virus 1, 2, 3. 3 strains are there. only HTLV-3B strain which causes HIV/AIDS. So similarly, we have taken the virus HIV-1 virus. We can take MT4 cells, and we have taken the MTT assay. MTT assay is a cell viability assay. Very simple. Already I told you, one virus can enter the cells. They will destroy the cells. Thousands of viruses can be released. So they cause a cytopathic effect. We have to quantify the CPE cytopathic effect by the cell viability assay. Simple MTT assay. This MTT assay used for the cancer studies also. If cells should be viable, they have the mitochondrial dehydrogenase. If the cell should be dead, means there is no mitochondrial dehydrogenase. Mitochondrial dehydrogenase is transfer the MTT dye into yellow color into blue color. Intensity of color directly proportional to concentration of cell. It is simple basic parameters. Like LD50 and ED50, we have to measure 50% concentration of drug which inhibit the virus, 50% concentration of drug which causes cytotoxic to the uninfected, non-infected cells. Like LD50 lethal dose, effective dose, like we have to measure the two parameters, and all the efficacy can be checked with the standard acetylthioamidine. Acetylthioamidine is a drug which is approved in cancer in 1963, which can be used for treatment of HIV. So HIV virus, we cannot be able to perform the ordinary microbiology lab. We need the PSL3, biosafety level 1, 2, 3, and 4. Like a coronavirus or Nipah virus or Hydra virus, we need PSL4. For HIV virus, we need the PSL3 facility. So we have to analyze by using the HDS, high throughput facility. When I did my postdoctoral position in Belgium, we have to be a high throughput screening. Fully automated ELISA system for reading the cytopathic effect, and multi dispensers for dispensing the medium and virus. And so we have take the microtiter plate 96 or 360 wells microtiter plate, one dispense with the medium and add the cell lines and the virus. It should be incubated for 3 days. One virus could multiply to 3 number of viruses in 3 days. We have to incubate 3 to 5 days. After 5 days, we have to stop the process, and we have to add the NTT assay, and we shake it well in a mechanical shaker, and we have to analyze in the ELISA reader. So we have taken this 96 well plate in the 2 different rows. This is a wells which is used for the HIV. This is yellow color means there is no effect. The blue color means is effect. Very simple process. The HIV will destroy the cells. HIV will kill the cells. The mitochondrial dehydrogenase is not active. If we are adding the drugs will molecules or a plant extract which destroy the virus, and preserve the cell from the cytopathic effect of the virus. Once the cells should be preserved, protected from the cytopathic effect of the virus, the mitochondrial dehydrogenase is there. This side will explain that this is a MTT assay, it is a tetrasols. With the help of the mitochondrial dehydrogenase, will open the ring, they will form the pharmazones. One cell should be viable, then only they will open the ring. If the drug have the effect means they will kill the virus, they protect the cell means they will preserve the mitochondrial dehydrogenase enzyme. They will open the ring, tetrasoline ring with pharmazone. Only the pharmazone they will form the blue color. Tetrasoline links with the yellow color. The yellow color remain means no effect. Blue color is there having some more effect. The intensity of color directly proportional to the concentration of the cells. So we taken this is a pharmazone. We have dissolved the pharmazone with the acetone something, you can get the blue color. So the intensity of color directly proportional to the concentration of viable cells. So we performed from this above principles, we performed one of my PhD students, they prepared the 2,3-diphenyl quinoxyl in small molecule in my laboratory. They requested to me, sir, we will test the molecule. So we sent the molecule for testing. But the same HIV1, HTLV-3B strain in MT4 cells by MTT assay, we calculated the 2 parameters, IC50 or EC50, 50% inhibitor concentration and a 50% cytotoxic concentration and maximum production. Maximum production is a power of the molecule which preserve the or prevent the virus-induced CPE, cytopathic effect in the MT4 cells. So from that, we have to compare the efficiency with the standard acetylthioamidine. So which inhibit the virus at the concentration of 3.8 microgram, and the CC50 was found to 15 microgram, and the maximum production was found to 59, whereas acetylthioamidine found to 0.0012, 65, 126. So this is a lead molecule. Now we developed further number of molecule. What we found that the developed molecule have no activity. We need a free amino group here for the essential for the anti-HIV So this is a small molecule. And currently we have used the concept of repurposing. You have to proverb old wine in new cup. Wine is very old, but cup is new. Similarly, this chloroquine is an old drug. With the help of our teacher, Dr. M. Chandramohan, we investigated chloroquine and from 1996 for the treatment of HPV and HCV by using the concept of old wine in new cup. By using the concept of old wine in new cup, what we have used viral drug is old. It can be approved for treatment of malaria, rheumatoid arthritis, lupus erythematosus, amoebic hepatitis. And also it's an immunomodulator. But we have to use for the new viral disease like chikungunya, dengue, and Nipah virus. Similarly, recently Trump also announced that chloroquine and their metabolic product hydroxychloroquine. Hydroxychloroquine is actually a metabolic product of chloroquine, which can be used for treatment of, which can be approved for the treatment of WHO recently for the COVID, SARS coronavirus. So we have investigated for the other virus also. We tested the chloroquine against the chloroquine, hydroxychloroquine and primaquine against the HIV-1 MT4 cells. What we found that 7.11 microgram they inhibit the virus. Toxicity was found to be greater than 33. They found to be 90% inhibition. Number of thousands of reports are available for the antiviral activity of chloroquine against the HIV and other human pathogenic viruses. Just a proof of concept, we tested the concept. Otherwise, hydroxychloroquine and primaquine does not act against the HIV from our report. Maybe other assay system maybe that may be active. So just we are using the repurposing of old drugs for new viral diseases. And they will come to the molecular target. Any small molecule which inhibit the HIV virus along with their molecular enzymes should be very effective. For allopathic medicines, we want to get approval for a particular drug. We want to know that the drugs which prevent the HIV virus and the drugs which prevent the any of their molecular target, HIV integrase or reverse transcriptase or protease. Already I told you in Hindu religion, Vishnu, Shiva, and Brahma is very, very important God. Similarly, for HIV virus, these three enzymes, HIV reverse transcriptase, HIV protease, HIV integrase. These three are very, very important for the multiplication. Currently approved drugs for treatment of HIV virus, which belongs to any one of the virus. So now we'll discuss about the HIV integrase. Is the enzyme which is present only in the virus. It is crucial for HIV replications and no cellular counterpart. That means they are present only in the virus, not in the human. And the low drug resistance. And it is an attractive therapeutic target for treatment of HIV virus. So we selected the therapeutic molecular target for our research work. Already, so we have long history. You know the very famous turmeric, which containing the curcumin. Curcumin which contains the 2 keto group as a pharmacophore. You know that pharmacophore means it's a part of the molecule which is essential for the designing of antiviral drugs. So here, curcumin which contains the pharmacophores, which is used to design the new generation. So curcumin which is tested for HIV integrase, which is also active because of the cellular toxicity, their molecular structural feature, which can be used to design this. So currently used drug like raltegravir, electrogravir, valtegravir, recent drugs which belongs to the HIV integrase inhibitor approved by FDA. They are having the viral clearance very faster when compared to the HIV reverse transcriptase and proteases. So they are two molecules which is designed based on the pharmacophore approach from the diheto group of the curcumin. So we prepared a number of molecules like A, B, C, D, and we tested for HIV integrase in collaboration with the Belgium. What we found that all molecules which have potent activity against the HIV integrase. So this paper is published in the, we studied both inhibition of overall inhibition of HIV integrase and I am already explained the 3' processing and strand transfer. We published the paper in the International Journal of Drug Design and Discovery Process. So these are some of the few examples. the same molecules which is prepared by us belongs to the isotope derivative. Before we tested the overall inhibition of HIV integrase, here we tested for the individual molecular mechanism. So we tested for both 3' processing. Already I told you it's like a double strand of HIV DNA, cDNA, which is coupled with the double strand human DNA for the integration process. So these are two steps involved: 3' processing and strand transfer. 3' portion of the viral DNA will coupled with the 5' OH of the human DNA. 3' following it. Then 5' portion of the viral DNA will couple with 3'. So what we found that all the molecules are chlorine, bromine, iodine, and N-acetyl substituents. We tested. What we found that both steps can be successfully blocked by this structure, ring. Next, India is one of the richest biodiversity country. We have lot of medicinal plants which is available in India, bound and powder. So we have to try to utilize. Recently, we have the successful history from the China. China is effectively control the COVID viruses by using the TCM, traditional Chinese medicine. So they are combined with the herbal medicine along with the allopathic medicine. They effectively control the SARS coronavirus, the Wuhan city instead of city of Wuhan. So India is one of the richest countries. This plant, Wrightia tinctoria, is called bedpale. In Siddha and Ayurvedic medicines can be used for treatment of psoriasis. And one more happy news: the drug which is approved for antiviral drug 65 for the smallpox virus and vaccinia viruses called as methicillin, which contain the isotyne thiosemicalbusis, first clinically approved drugs belongs to the isotyne derivative, is isolated from this plant. So we tested this plant extract, various extract, chloroform extract, methanolic extract, aqueous extract, ether extract, and many extract we tested in the both steps of HIV integrase. This work performed with the NIH, LMB, Laboratory of Molecular Pharmacology, NIH USA. So what we found that AWT aqueous extract having 1.9 and 1.4, which is effectively control the activities of HIV integrase. This is for published in the indexed journals, Antiviral Research 2010. And I already told that any small molecule or extract which inhibit the replication of both virus and their therapeutic target, then only it can be approved for the treatment of HIV viral diseases. HIV viral diseases. So we have taken the various extracts which is tested against the antiviral activity against HIV1, HTLV3B in MT4 cells by FTT assay. So what we found that we are comparing the maximum protection. So chloroform extract having 48% maximum protection against the replication of HIV1. Then we are using the bioactivity guided fractionation methods. So for the separation of active molecule from that. So we isolated isatine, indrubin, indicotin, triphanderin, many molecules from the chloroform fraction. We tested against the data not shown. We tested against HIV1 virus replication, the ELISA method. What we found that indicotin is a molecule which is effectively control the virus replication. This is also paper available with Indian Journal of Pharmaceutical Sciences 2006 or 8. And then you heard the name. We had funded research from the Noni Biotech Limited. We have prepared the different fraction of Noni. Noni is simply called as Morinda citrifolia, Nuna. Morinda citrifolia, which can be effectively used for treatment of many diseases. So it's one of the traditional Indian mulberry. So we have the foundation, World Noni Research Foundation, Noni Biotech. They funded tested the Noni extract. It's acetone, AMC acetone, fruit methanolic, chloroform, ethyl acetate, hexane. What we found that this extract having excellent antiviral activity, HIV activity against the HIV integrase. And the acetone extract having 0.031. And we found to be the acetone extract also inhibit the replication of HIV1 in MT4 cells. So this is also published in one of the famous journal, Antiviral Research 2010. We explored the two medicinal plants, Wrightia tinctoria and Morinda citrifolia. First time in the world of literature for HIV integrase inhibitory activity. Once again, we are using the concept of repurposing. It's like a proverb: old wine in new cup. Wine is very old. If you are keeping wine year together, what happen? We have wine power, that power should be more. So similarly, a drug like a chloroquine and hydroxychloroquine and primaquine, they are approved for treatment of malaria, but we can use for treatment of many viral diseases, especially for chikungunya. Chloroquine and hydroxychloroquine are effective. Similarly, what we found that primaquine is act as a lead molecule for inhibition of HIV integrase. Primaquine will inhibit the enzyme HIV integrase. We prepared the 3 more derivatives from the primaquine and we tested for integrase. Next, very interesting things. So, because HIV virus, so far there is no effective drugs or vaccine. No effective vaccines are available. So, with the research of International Vaccine Action Initiative Group, IAVE, nowadays they reported simply told that they stopped all the efforts for the development of vaccine for HIV. It is useless. So only available options would be ART, antiretroviral therapy. That also, drugs came 5 to 6 years, the HIV will get mutated. So ultimately, we can go for new targets, new molecular therapeutic target for developing the new drug molecules, small molecular entities to minimize the drug resistance. So that's why we selected the HIV integrase, and we selected not only HIV integrase. We found that virus can replicate in the host cell with the help of the human proteins. So we selected the protein called as LEDZF, lens epithelium derived growth factor, P75 transcriptional cofactor, and we confirmed that integrase will bind with the LGF for human host protein, then only it can be replicated. If you remove the LGF from the host cells, add the virus, there is no replication. So from the proof of concept, we confirm that LGF is very, very important binding partner of HIV integrase. So we developed the alpha screen assay. So this is a special assay, donor and acceptor, 2 beads are there, donor and acceptor. One bead which containing the HIV integrase, another bead which containing the LGF. So both beads come together, they will emit some fluorescence. If you add the drugs which prevent the binding of HIV integrase and reverse transcriptase. So one of the challenging thing, developing the protein inhibitors in the fermenters is very challenging. They are using the alpha screening assay, the alpha proximity assay. So we try to use that assay. Actually, this can be used in the cancer research. We have to use in this assay. One bead we can add the integrase, another bead we can add the host protein. Then we will add one without drugs, one with the small molecules. So the drugs which prevent the protein interactions is very challenging. That changes the emission. So this is assay. It is alpha screen assay, it's a proximity based assay. They have the protein interaction. This is a photosynthesizer, this emission is there. This is a special type of assay. We have actually it is used in the pharma industry for designing the anti-cancer drugs as a protein inhibitors. So in order to validate the protein interaction assay by the alpha screen methods, we have taken the HIV integrase inhibitor. Already I told you there is 3 drugs are currently available: raltegravir, eltigravir, and valbogravir. So we have taken the eltigravir as a standard. What we found that they inhibit the HIV integrase and LGF interaction at the concentration of 1.6 microgram per ml. So we taken from the Journal of Medicinal Chemistry as a one internal. So for developing the new assay, we have taken some internal standard. For anti-HIV activity, we have taken the acetylthioamidine, which is approved for treatment of HIV/AIDS. We have compared the activity of our molecule or plant extract with the acetylthioamidine. So this is our 2010. This is publication from our postdoc laboratory. So they developed the world first time they developed novel therapeutics for quinoline derivatives. So this is a paper will appear in the reputed journal like Nature Chemical Biology in 2010. So Nature and Science, this article will publish the newest innovations like a Nobel Prize capacity. So this is a paper published in 2010 by the our boss. Then I decided to develop some small molecules. So with that, I developed a small molecule quinoline derivative. So which inhibit the HIV integrase LGF interaction at concentration of 10 micromolar. Then we performed the So it's a drug research like a sambar. You heard the sambar. Sambar, if you want to improve the taste of the sambar, we have to add the more vegetables. So similarly, the drug discovery process is a chemistry, microbiology, virology, biochemistry, clinical sciences, physician. Like it's a multidisciplinary approach. We need the multidisciplinary task to discover. So we have to use the bioinformatics. So we are using the bioinformatic techniques. It's called CAD. It's a computer-aided drug design. Already I told you the HIV having the active site having the catalytic triad. Active site is nothing but it's a big elephant is a big size, but a small iron rod will control the elephant. So similarly, protein is a macromolecule is a big size. Among that, three enzymes which is present in the active site. It's a small cavity-like appearance. It's like AAS, aspartic acid 116, aspartic acid 64, glutamic acid 152. So these three catalytic triad is a very important for the interaction of drug molecules with the ligand with the active site. So we perform the Schrodinger software, we perform the small molecule binding with the active sites. What we found that is bind strongly with the active site of the enzymes. Then we have to use the pharmacophore approach. Pharmacophore approach, we are superimposing the molecule with the drug interaction. So what we found that what kind of interaction they form, what kind of amino acids they will interact with the active site with the residue. Then we have approached the pharmacophore. So this is a pharmacophore model we developed by superimposing the molecules. We developed the pharmacophore, common pharmacophoric model. Then with the help of the pharmacophoric model, now we are screening 40,000 molecules by using the library from the zinc database. We are getting more active derivatives. So this computer-aided drug design will minimize the time. Will minimize the time of drug discovery process. Speed up the before they need 10 years for developing one molecules, but nowadays within a day we design the more molecules. So I want to show some of our research publication. So we are working on the novel therapeutic approaches, HIV integrase and inhibitors in collaboration with the University of Southern California, Los Angeles, Nauri Nimati. We have prepared the molecules, which is isotope compounds, which is tested against the HIV integrase and LZF simultaneously. So they will prevent the both redas processing and strand transfer, and only two molecules having capacity of dual mechanism. They will inhibit the HIV integrase as well as HIV integrase LZF interaction, sp3-5H MEA. Unsubstituted without bromine, chlorine, or methyl, unsubstituted nucleus is having more binding interaction. So this paper is published in the International Journal of Drug Design and Discovery. And we developed a small molecule like a very small molecule like a 2-amino-4-phenyl thiazoles, 2-amino-methyl pyrosoles, these drugs, and in collaboration with the East China University of Science and Technology, by using the CAD as well as biological assays, what we found that this is a small molecule which inhibits the HIV integrase and LZF interactions. So we have so far there is no drugs approved under the category of inhibition of HIV integrase LZF category, but we have taken the standard from the nature chemical biology molecules. The small compound 3 from the nature chemical biology are found to be 11.99, almost 12 microgram, and the small molecule 2-amino-4-phenyl thiazoles small molecule which inhibit the 12. We will confirm by using this docking studies also. They have the glide score. They have the glide score of 5.46, 5.86 like this. And also we confirm further the quinosolines. Nature group they reported for quinoxolone derivatives. We tested for quinosolone derivative. What we found that the nature compound having the IC50 11, our quinosolone molecules having 10.08 microgram. This is more potent against these. And already I told you we have the funded project from the Noni Research Foundation, Chennai, 15 lakhs foundation. We investigated the complete profile of Noni fruit juice extract under the isolated compound for the HIV. So we are the first group in the world. We reported for the inhibition of HIV integrase activity by these fractions F1, F2, F3, F4, F6 fractions inhibit the process, as well as the isolated compounds. We found that isolated compounds C14 methyl fractions was found to be inhibit the HIV integrase as well as both the enzymatic process. last but least, I will express my thanks to our beloved Professor Eric D. Clark. He is the inventor of HIV virus. 1984, we developed the assay from the Rega Institute for Medical Research, Belgium. He is the first person to explore the antiviral activity of acetylthymidine, AZD, acetylthymidine, for the treatment of HIV, And also recently he explored the drug like a tenofovir, adifovir, a trifoxylate derivative for complete treatment of HIV. I did my postdoctoral training. Thank you, sir.
Hello.
Questions in the chat box, sir. Anyone can read, we will answer. Yes, sir. Yes, sir. Question, just read one by one. Hello? Hello? Can you answer the question? One question from Sumaya. Okay, is audible, sir? Is audible? Is audible? One question.
Selvam, one question is there actually.
Yeah, sir. Many questions are there. I'll answer one by one, sir. Okay.
Just.
Number one question from Sumaya. Patient relying the antiviral drugs is a free viral RNA or DNA from the dead cell or at least the serum. It's a good question. We have one phenomenon is called as empty surface antigen syndrome. Even the virus could be dead. Their virus particles will be circulated sometime in the blood. So for complete removal of viral genetic material from it will take 2 times. So for that purpose is the best way. We are doing the real RT-PCR. Currently using the COVID or SARS coronavirus, they are using the RT-PCR. Similar way, we have to use the RT-PCR and viral load for the confirmation of the viral infections. Especially they are using the nanostring as well as RT-PCR, real-time PCR, RT-PCR. We have to quantify the number of viral particles. It is the best question. Number one. Number two, noni-mediated nanoparticle effective for HIV virus, sir. So noni having generally the Morinda setifolia seed fruit which having the powerful anti-inflammatory as well as powerful antioxidant and also it contains lipopolysaccharide having the powerful immunomodulatory. We documented noni acetone extract as well as the noni extract which inhibit the enzymes HIV integrase. So we can use the noni for the, we have to use it is very somewhat toxic to the system. So we have to use the other biodegradable nanoparticle like a chitosan, albumin nanoparticle for the HIV patients. Next question, can you use the Computational software for calculating the computational effect of drug antivirals? So best choice would be either Schrodinger Clyde score, or we are using the Discovery Studio. Asli Discovery Studio is the best way. If you want freeware, we can use the SwissDoc. So www.swissdoc is the best software for the drug design purpose. As well as Autodoc Vina also is a best way. But the golden standard, either we are using the Schrodinger upgraded version, or we have to use the Accelerate Discovery Studio. Next question from the Bhaskar. Please discuss the Siddha medicine for treatment of coronavirus like this. So in Tamil Nadu, you have the history of Kapasura Pudini. Kapasura Pudini is containing 15 phytochemical constituents. Currently, we are using the government of Tamil Nadu is also approved by the Ministry of Ayush along with allopathy plus. And Kapasura Pudini or Nilavembu Pudini is containing andrograph is calculated like this, 15 ingredients. My YouTube also available. Please refer my YouTube, my presentation to YouTube. We have clearly explained use of Kapasura Pudini and Nilavembu Pudini for the treatment of management of COVID along with the approved drugs. In Chennai also, daily 3,000 to 4,000 patients are get relieved by in combination of this allopathic drug along with the Siddha system of medicine. Next question is, noni-based nanoparticle will improve the drug delivery system effectively, when especially for the you have to target for the delivery of the nanoparticle to the T cells, lymphocytes. That they will because the viral target is a lymphocytes. This is the best way. Next question, excellent person. Please avoid the. Okay, just don't take mistake. So in order to understand the betterment, okay. If a Christian also, Muslim also, whatever gods are important, all are equal, all are important. Just I will explain things. I not promote anything. All humans are equal, all gods are equal. All like Vivekananda said, all the river goes to sea only like this. So India said, just I will explain that things important. Okay, don't take mistake. And any other question? Why I am taking that HIV? This is only an uncurable disease. They are going to be a lifelong up to the end of the world. This is going to be a endless problem for a human being. And nowadays, everybody focusing mainly on the corona. Like this, HIV also now very important. Our 0.5% population suffered by the HIV, and so far there is no effective drugs are available. So my research last 15 years focusing on the invention of herbal-based medicine for HIV. Because India is one of the richest biodiversity country, we have lakhs and crores of medicinal plants are there. And we explored the Morinda citifolia noni as well as Wrightia tinctoria for inhibition of HIV integrase inhibitor world first time in collaboration with the National Cancer Institute. Any other question from Principal Sir?
Selvam, it is nice actually because all the participants, they put a lot of questions, I think. So nothing more specific, Selvam.
Yes, sir. So we have to motivate, sir, motivate the student to do the research, future students, we have to motivate. That's why this society is established. So we have conducted many programs to motivate the students, to produce the next generation leaders.
Yes, sir, it is required.
Please activate the Hassan Muhammad, Isaiah Muhammad, is from Sudan. Please can you activate that connection of Hassan Isaiah Muhammad? One person from just video showing from Sudan.
Raising hand raising.
Hassan?
Please raise the hand, sir. Please ask them to raise their hand, sir.
Hassan, please ask to raise the hands. Hassan. Hassan, raise the hand. Raise the hand.
You know,
I
It's
think.
from Professor Hassan Isa Mohammed from Sudan.
yeah, you only know.
Okay, sir, you please go on.
Selvam, go ahead.
Okay, okay. Hassan.
Hassan, Hassan, sir, please go ahead, sir.
Hassan. I think it's not able to listen simply. Okay, madam. If not audible, please come. And any question from faculty members? Any question from faculty member?
No questions in matter.
Okay, sir. That's all about the question, sir. Thank you for the session, sir. And thank you for motivating us. You have explained and discussed about the virus basis, category of viruses, structures. And apart from life cycle of that, and you have targeted the you have explained about the molecular targets also. And thank you for the vast information you have given. So as you said, apart from corona, we are discussing all these days about the corona only, but it is the most important, as you said, 0.5% of the population of India is affecting with this. So, and you are doing research using herbal based medicine. So thank you very much, sir, for the explanation given by you. Thank you, one and all.
Okay, thank you, Selvam sir.
Thank you, sir. Thank you, sir.
Okay,
Thank
I
you,
will
sir.
talk to you later.
Okay, okay. Madam, can you upload YouTube, madam, video?
Sure, sir.
Are you upload? Madam, can you upload this video? Video you have to upload after this chart, live chart. That video should be uploaded now.
It's going to upload, sir.
Okay, okay.
It's
That can be.
uploaded, sir.
Okay. Regarding the nanoparticle.